Hi, everyone. I'm Stephanie again. And in this section, I'll be talking about reading NWB data from DANDI, which I expect all of you all will be doing a lot over the course of this week. And so to get started, if you want to follow along, this will be kind of working through different notebooks. In the first part of this talk, I'll go through Python, using Python to interact with NWB files with PyNWB. And then in the second half, I'll talk about using MatNWB to interact with NWB files in MATLAB. So you can download the notebooks from this link if you want to follow along. They should also be available in that slides resources page that was shared in the Slack channel. So I'll give people a few seconds to go ahead and do that. And then, like I mentioned, if you have Dandy Hub set up, I'll show you and demo how to run it through Dandy Hub, but then you can also always just run the notebook locally in a Python environment or MATLAB account.
So download the notebook, the IPYNB?
Yeah, we'll be going through the Python one first if you want to download that one. I see people still looking at the screen. It's also, like I said, in the slides folder, so I'll give people a few more minutes. Let me show you. On the Slack channel, in the bookmarks, we have the slides link. And so the notebooks are also here, if that's faster. All right, and so if you are interested in using Dandy Hub, what you can do is go to hub.dandyarchive.org, and that will open up, and if you've already started, it'll open up an instance, or it'll prompt you to log in and start a new instance. You can start the base environment. There's also an option, as Ryan mentioned, to use the GPU. And so it should launch you to this kind of notebook. And so if you want, or this kind of view. And so if you want to upload that notebook that you just downloaded, you can click upload files. I had already uploaded it earlier. And close that. And then you should have this kind of notebook set up in the Dandy Hub. And then Dandy, if you have used Dandy Hub recently, they've changed how some things work in terms of environment management recently. So I'll just kind of briefly give an overview of how to set that up. There are also instructions in the notebook, but basically you'll go to Services and Environment Management. And then you can create a new environment for your user in this package manager. You might also have to re-log in here to Condastore. Yeah.
So
yeah, go ahead.
we can do this only if we already set up the Dandy Hub?
Yeah, so if you're not able to log into Dandy Hub yet, you can open up the notebook with a local Python environment, and I'll provide information on how to install or which packages to install.
And what
Yeah.
is the...
So, is it, because it says in the Dandy Hub, it says otherwise sign in with your GitHub account, and then there's a button that says sign in with key code. Is that the one?
You want to use the GitHub account.
The first button is G-Code, then that will take you to another one that says GitHub.
Oh.
Yeah, all right. The Dandy team changed everything last week.
Yes.
He needs this.
I signed in with my GitHub account. I ignored the keycloak thing, but
Yeah, I'm also getting that same with the GitHub.
So what probably is happening is the Danny team needs to approve some of your registrations, and that isn't happening in real time right now. I would suggest going with the second approach.
Yeah.
Doing it locally, not on the other.
Yes, I'll just show maybe, it seems like Dandy Hub might have issues we might want to go through. How many people are using Dandy Hub currently and getting it to work? Okay, so I think I'll focus on a local environment installation, and then maybe we'll set aside time later if people want to get set up with the Dandy Hub instance.
I have another question.
Yeah.
I have never in my life used either Python or Jupyter. So am I expected to install Python right now for what we're going to do here? Or can I just jump it and there's going to be a MATLAB?
Yeah, so this tutorial will assume that you have Python installed and you can set up in a virtual environment. We had talked about maybe having an optional session later about kind of Python best practices and getting set up on your computer, so we could talk about that. But I would say for now, maybe follow along with the tutorial, or just like follow along, I'll walk through, and then the MAT NWB one might be more relevant to you if that's what you're currently using. Yeah, I didn't say.
If there's going to be a MATLAB NWB one, I think I'd like to just follow along. Sorry, and then.
Yeah, because you don't have Dandy Hub working, right? Because that's the other option. It's fine if not.
Possibly, I might soon.
Okay. If you can get Dandy Hub working, that will have Python and set up in the environment we can set up easily.
Just a quick survey. How many of you all plan to use Python during this workshop? Okay, and how many plan to use MATLAB? Okay, so we can have a separate MATLAB session.
Yeah. Cool.
But for now, just hold tight, and you can follow along this watch, and some of the same concepts will be in both.
Yeah. Cool.
Could you remind how you got there?
Yes. So if you are interested in this, these slides are also in the slides folder if you want direct links. But I downloaded the notebooks from the slides folder from that link, and then I went to hub.dandyarchive.org. Where did I have that pulled up? Okay. All right. So I had already set up a Dandy Hub environment, so I'll use that to run things. But basically, if you are running this locally, or if you were setting up the environment for the first time, we would first need to install these packages. And is this size good for everyone? I can make it a little bigger. Yeah. So if you're running this notebook locally, you'll need to install these packages: Dandy, PyNWB, RemFile, and Matplotlib using pip or whatever your favorite Python package manager is. And then if you're running on Dandy Hub, I've provided instructions, but it sounds like maybe it's not working right now, so we can go through that later. And yeah, to take a step back, since we ran into some technical issues, I'll be going through reading and exploring NWB data from Dandy. So the goals of this section are really to learn how to download data from the Dandy Archive, to read and explore an NWB file, do some basic visualizations, and then most importantly, especially as data sizes get bigger, how to stream data from the Dandy Archive so you don't have to download entire files at once. So how are people who want to follow along? Can I get a thumbs up if you have the notebook open and at least can maybe click through cells if you have the environment set up?
If you don't, raise your hand.
Cool. All right, so after you've set up your Python environment, we're going to start by importing these packages that we installed. Like I mentioned, really RemFile, PyNWB, Dandy, and Matplotlib are the main ones we're using in addition to NumPy. And in this notebook, we're going to be working with NWB data from a session of an experiment by Chandravati et al. And so in this study, the authors recorded single neuron activity from the medial temporal lobes from human subjects while they performed a recognition memory task. And so this on the DANDI archive is going to be DANDI set number 4. So if you have the notebook open, we can go ahead and click that link to see where that is. And so like Ryan showed earlier, this is a view of a DANDI set. You can see there's a description of the data that's contained. There's information about the data contributors, funding. Sometimes people will link to code, resources, or the paper in this related resources section. And then there's also a summary of some of the information contained in that file. And so when you're starting to download data from DANDI, there's a couple different ways that you can do that. And so the first kind of simplest way to get started is to use this GUI web interface. And so to do that, if you go to Files, and we go to, say, example, the subject, there's this little download button where you can download the asset, and it'll go to your downloads folder. And so if you are working on DANDI Hub or in that notebook, you'll want to move a file that you're interested in analyzing to that specific folder. And I think the one we're going to be using actually is subject P11 HMH. So you can download that and move it to the appropriate folder. I'll leave it up here for a second. Yeah.
So I'm running the notebook locally, and I have this arrow on cannot import read NWB from Pi NWB.
Okay. So if you have an older version of Pi NWB installed, the read NWB function is a newer one. So you could do pip install with -- upgrade, depending on what you're using. Let me see if I can type that up. And maybe Ryan can assist more directly. But yeah, this notebook does assume you have one of the later versions of Pi NWB installed. So if you have been using it before, you might have wanted to set up a fresh environment. Okay, so as I mentioned, one of the quickest ways to download files from DANDI is to just go into that file and click download. However, in many cases, as you start to get more in depth into your analyses, you might want to download multiple files at once, or all the files from a subject, or all the files from a DANDI set. And so DANDI has a couple of options that you can use to do that. There's the DANDI command line interface and the Python API. So first, I'll go through the DANDI command line interface. And so here, if you go back to your launch page and we open up a terminal, and I think I'm going to need to activate the environment I want to use. If you have a terminal and you're in your environment, you can do DANDI download and then DANDI, let me make this bigger, with the name : and then the number of the DANDI set that you're interested in downloading. And so if I click run, what you'll see is I'll start to get a bunch of files being downloaded because it's trying to download the entire DANDI set. So I just aborted that temporarily, but just to give you a sense of how this works, if you do want to download all of the files from a DANDI set, this is a really easy way to do so. It'll list out all the files, give you information about how far along in the process it is and the status, and this can be also good if you ever have internet connection errors. It'll kind of pick up where it left off and continue the download process. So this can be a more robust way than the web interface. Another way that we can use the DANDI command line interface is you can actually just specify the web link straight from the DANDI archive page. So if we're here and I just copy this URL, I can do DANDI download, and I think I'll have to remove this page at the end, but this should get all of the information for that subject, for that DANDI set. So I'll do DANDI download, and it's downloading that file, and now I have it on my computer. So you can grab that URL. It's a really quick and easy way to download with the command line interface. You can also, on the DANDI Archive, there's a way to look at the asset metadata, and Ben will show you later more also advanced ways to query the DANDI data. And so this will give you information about this can give you a content URL, and so you can also use that to download data directly. And what that content URL is coming from is basically whatever link is here if I copied it when you're downloading, this link here when you're downloading data straight from the web user interface. So to give an overview, if you're using the command line interface, you can download an entire data set. You can copy and paste the URL from the web app, or if you have the asset link, you can download a specific file. And the DANDI docs have more information as well. And then finally, if you say want to have DANDI files downloaded as part of a Python script, you can use the DANDI Python API, which basically mirrors the command line interface. And so here I'll demonstrate how you would download using the DANDI set ID and file path. So we have our DANDI set ID here, 000004, and then our file path, which was also shown here. We have our subject, and then here was the name of the file path that we're accessing. And so with the DANDI API client, we can get the DANDI set information, we can get the asset, and then download it. And I have it set up here where if it's already, oops, I didn't run. Let me run the import, okay. There we go. And I had already downloaded it to my folder, so I skipped it.
Is there any way to use the DANDI API client to look through the DANDI set IDs, to find these subjects and files?
Yes. Yeah, there's definitely more ways to get a list of all of the assets and subjects available within it. I don't know the exact commands, but I think they have more details on the DANDI API website. Do you know of them off the top of your head? Or
I don't remember the exact command, but
Yeah.
It's documented on the website.
Yeah.
And,
let me see.
in my talk, there was a link to simple tutorial and advanced tutorial, and those can go through searching programmatically.
Yeah, so if you're interested in using the Python API, there's some API documentation here, and that should give you a lot of different options for exploring the assets. Oh, a specific part of an individual NWB file. I think we'll talk a little bit about streaming specific parts of an NWB file if you don't want to download all the data, but with the DANDI client, I think you're downloading entire files, as far as I know. Cool. Any other questions? So that's kind of the overview of the three main ways you'll download data from DANDI. Any other questions about any of that? Cool. So the next part, now that we've downloaded our file, I'll demonstrate how to start reading and exploring an NWB file. And so, like I mentioned, this read NWB function is new, but it's kind of our simplest way to just get started really quickly with reading an NWB file. So here I'm specifying my file path for that file I'd previously downloaded, and then read NWB. And then if you're in a Jupyter notebook, there's a really nice interactive representation that you can get of the contents of the file if you type NWB file for that object we just defined. And so here you'll see this kind of broad summary of all of the different components and all the different data contained within this file. So information like the session start time, related publications, the creation date. We also have all of our data contained kind of within these different subgroups. You can think of it kind of as subfolders within this root NWB file structure. And so under the hood of that read NWB file, the kind of main class that's carrying out the NWB reading and writing is the NWB HDF5 IO class. And so NWB HDF5 IO is reading and writing data that's in the HDF5 file format. And so that's a really popular hierarchical storage format for scientific data. And so a lot of NWB files that you'll see on the archive are going to be in the HDF5 format. Another backend option that's available is the ZAR format. I don't think there is that many of them, or if any, on DANDI currently, but if you're interested in that, we have tutorials and we can talk more. And so if we want to do more complex queries or complex ways of reading in the data, or sophisticated really, we can use the NWB HDF5IO class. And so here we can specify the file path and then the mode. So other options here would be to append to the file or write if you're writing to a new file. And so we'll define our IO object, and then you do io.read to read in the NWB file. And this will basically look the same as the NWB file we previously read in. Another way you can use this IO object is as a context manager. So the advantage of that is that with a context manager, the file will be automatically closed whenever that context finishes. And so in Python, that's designated with the tabs or indents. And so with this method, if you have with NWB HDF5IO as IO object and read it in, the data will be accessible within that context. And then once you unindent or exit that context, then the data will no longer be accessible. So you might see this in a lot of tutorials, and it's a common thing that people trip up on is just trying to access the data when the context has been finished. So here I'll run it here, and it'll do basically the same thing. All right, now that we've read in our NWB data, I'll kind of walk through different ways that we'll access the data within the file. And so we have NWB file.subject if we want to ever get information about the subject used in our experiment. So this will give us information like age, sex, species, et cetera, and the subject ID. If we're interested in stimulus data, we have the NWB file.stimulus section. And so here you'll see if we print out what's in this structure, we'll have this, it's a Python dictionary, and it contains PyNWB objects that are representing different types of data, in this case, images. So here we have a single key value stimulus presentation, and it is a PyNWB optical series object. And so in this case, it's an RGB image. And so if we explore that further, if we want to access that, we'll do NWB file.stimulus, and then we'll use the brackets with classic Python dictionary accessing to get the stimulus presentation. And now we can see, again, this kind of more interactive representation printout of what's specifically within the stimulus presentation optical series. So we'll have information about the data, the shape of the data, the size, different compression options, the timestamps. And this is another way to kind of explore more in depth within the file. All right. Now, one thing that's important to note is that data is lazily loaded with PyNWB, and so data arrays are read passively. And so what that means, if you're not familiar with, this is also used in NumPy commonly, if you're not familiar, so if you do stimulus presentation.data, what that's returning is not reading in the data values, but it's presenting this H5Py dataset object that you can then index into to read the data. And so it's not loading all the data into memory at once. Let's say you have a really long electrophysiological recording. You don't want to read in all of that data at one time. And so you can access it selectively. So if you want to read the whole array into memory, you can do stimulus presentation.data bracket and colon, and that will give us, then if we print out all stimulus data, we'll see no longer, it's not this H5Py dataset object. We're actually reading in the values of the array. Yeah.
If we see this shape, how can we interpret and understand what kind of stimulus is provided? Is there a way to actually understand this number?
Understand the numbers of the array or the shape you're saying?
What stimulus actually happened and what are the actual stimulus? Because I couldn't see the descriptions when I see the information. wonder, so I wanted to understand what data set it is.
Yeah, so the question is understanding the array data, or like what is the data type that's being represented in this. And yeah, so it's this optical series data type. So there will be defined by the schema some definition of how the data should be structured and formatted. So in this case, it's going to be an image, and it's going to be this will all be defined in the schema. So it'll be the first dimension is timestamps, and then it's X by Y coordinates, I think. And then the last, either it's a 3D or a 4D image where the last dimension is RGB values, or it's a grayscale image. And so that information about what types of data might be contained will be defined by the types that are defined in the NWB schema, if that makes sense. And if you want to explore more, we have the PyNWB documentation. And so if you want to know more about specifically that optical series object, then the API documentation might be helpful. And so look, optical series. This is going to give us information about the array. So it's going to say images, maybe 3D or 4D. And so the API documentation, in addition to the schema documentation, can be a useful way to understand what's in the file.
So how is it managed in the rack when you are indexing HTML5 objects? Are you only loading on the rack that subset or that slice of the data? So if I have a loop over time, for example, and each time I'm calling data and I'm getting a specific index of that, is only that time point loading on the rack and not the entire file?
Yeah, so it'll slice into the file. That's basically what's happening. I think the exception would be if the data is compressed in any way and chunked, then it would load, the data might be chunked into different parts and it'll load the entire chunk at once. But I think slicing into I actually don't know how H5Py works under the hood for that part. But
Right. So it will load just if the data are not chunked or compressed, then it will, if you use square brackets to pull out index 5 to 10, it will only load into RAM, 5 to 10.
yeah.
And if the data are chunked and compressed, it will load the chunks that encompass that slice. So it's the smallest amount that you can load. Yes.
Yes. But yeah, it's very, very useful because a lot of times you might maybe want to only get the data around a specific time point or certain events in the task. And so you can slice into a subset of the whole electrophysiological or optical array and get those time points. Yeah. And yeah, did that answered your question with the, okay, great. And yeah, so here, like I showed earlier, you can kind of explore different parts of the stimulus presentation data so we can get information about the shape, which if you look at the schema will kind of provide more context as to what is contained within it. And then, yeah, we basically already covered this, but with large data sets, it's often preferable to read in only a portion of the data, and so you can index and slice into the data just like if you were working with a NumPy array. And so if we want to, for example, look at a single image of the stimulus presentation data, you'll have stimulus presentation.data with your frame index. In this case, we happen to know that the data in this file was not stored RGB, but BGR, so we'll flip it. And then if we plot that data, which can also give us some information about what's contained, we'll see this image that was presented as a stimulus for this experiment. So another data type that you might often see or work with within the NWB files is the units object. And so if we do NWB file.units, that's going to give us a lot of information about the spike sorted single units that were recorded from in a single session. And so it'll store metadata about each unit in a tabular form where each row is going to represent a unit with spike times and then any additional metadata that the data contributors might have supplied. And so here, if we preview this interactive representation, we'll see we have this ID, original cluster ID, information about the waveform mean, SNR, and then the spike times in the electrodes. And so the units table is a special case of a dynamic table. And so these in NWB files, you can load easily into a pandas data frame if that's a format that you're more comfortable working with. And so if we do NWB file.units.to data frame, and then get the first few columns, you'll see that this has been converted into a pandas data frame, and so it can be used for querying, filtering the data in different ways. And then if you want to access, for example, the spike times of the first unit, what you would do is units data frame and then brackets with the spike times column and then 0 to access the first row of that data frame. And then one important thing to note is that in an NWB file, all seconds or all timestamps are supposed to be stored in reference to the session start time. So the data should already be all pre-synchronized and aligned, so that whenever you're seeing seconds, you should know those are all relative to the session start time. And I don't know if I mentioned this at the beginning, but an NWB file in general should contain all the data and metadata from a single session, so that you'll have multiple NWB files for multiple sessions. All right, so then to demonstrate one way to visualize spiking activity and to kind of interact with the different parts of the NWB file, here I'll show single unit spiking activity relative to when an image stimuli is being presented to a subject. So we'll iterate through the first 3 units and then compute the spike times relative to the stimulus onset and then create a raster and histogram. So here you can see we're accessing the units object. We're getting the timestamps from the stimulus presentation with this get timestamps method. And then if we run this section, we should see a nice plot with the rasters and histograms of the peristimulus interval. Another object that might be really useful, or section of the file that might be really useful for you all, is a trials table. So trials are stored as a time intervals object, which is a subclass of this dynamic table. So again, these dynamic tables store metadata about the experiment in tabular form, in this case about the trial specifically. So each row will represent the trial, and then you'll have the start time, stop time, and any additional metadata. And so there's a whole tutorial on working with these intervals tables, but here I'll just demonstrate that similar to the units table, you can do trials.to-data frame. And now we'll see that there's all this information about start time and stop time, as well as all these additional columns that the data generators added for the experiment. And then one thing I found helpful is a lot of times these column names are relatively interpretable, but if you want additional information, they should also each have a description that will give kind of a further information about the data that they contain. So for example, trial stim phase description will give information about the learning and recognition phase during the trial. And then trial data, again, we can map that back to interact with other parts of our NWB file. So here, let's see, we can visualize the stimulus images by trial. And so in this case, in this NWB file, we have the stimulus on time column that indicates when the stimulus is being presented. And so we can go back to that stimulus presentation object that we created from the stimulus section, and we can first check that all the timestamps seem to be matching up using an assert statement. And then we can filter the data frame and just get categories that match this landscape category, get the stimulus onset times for those, and then get the images where those occur. So if we look at the first 3 stimulus presentation images with this category landscapes, you can print them out. We can see how to access the data and kind of coordinate across the different parts of the file. All right, so that's kind of a brief intro into visualizing an NWB file. Like Ryan mentioned earlier, there's lots of tools and analysis and visualization tools, including NeuroSift, that can allow you to explore the file pretty easily. And then in this last section, I want to talk about streaming NWB data. So especially as data sets are getting bigger, and we're requiring more and more data for a single session, in a lot of cases, you might want to work with a really large NWB file or subsets of data from an NWB file, and you don't want to download the full thing. And so with this, with the streaming approach, you can stream data directly from the archive, and it lets you read specific sections of the file with and specific sections of the individual data files from archives such as DANDI. And so there's a whole PyNWB streaming tutorial where we have multiple methods, including FSPEC, if you're familiar with that. Today we'll focus on using REMFile, which was developed by Jeremy Magland, who also did the NeuroSift, and it's really optimized for streaming NWB files from DANDI. And so just to demonstrate how easy it is to stream data, here we'll again specify the DANDI set ID in the file path, and we're going to get the S3 URL of the NWB file stored on DANDI. So DANDI stores the NWB files in S3 buckets, and so we can get this S3 URL, and if we print it out, we'll see the link here. And this is what we can use as an input to REMFile to stream data from DANDI. And so, yeah, like I mentioned, REMFile is pretty simple and fast package, and it's really good for that initial load and access of NWB data. It is a relatively newer project, so if you want more specific caching capabilities, you can use packages such as FSPEC or the ROS3 if you want to use the HDF5 specific ones. And so here we'll take that S3 URL and we'll create a REMFile object. We'll then use that to create an H5Py file object. And then we can use that same NWB HDF5 IO and specify use the file argument to create a new IO object, and then io.read to read in the data. And so you can see we can still get that really simplified interactive representation. It looks very similar, or it should look the same as to what we looked at before. And then we can work with the data in the same way as when we downloaded the full file, but now we're streaming all of that data on demand. So we haven't downloaded the data, we're just accessing the different parts that we're interested in. And so for this example, I'll show we're going to specify a single frame of the stimulus data array, and we're going to print it again. And so now we've been working and we're just streaming this selective part of the file. And then it's good practice in general to close any files that you've already opened. So do io.close and iostream.close. And you can see how easy it is to just kind of quickly switch between streaming and downloading the data from DANDI to open it. So hopefully as you all are exploring maybe large data sets, you can start by streaming if you don't want to download the full file and just access the data in that manner. Any questions about the Python part of this tutorial? Yeah. Yeah, there's different ways to set up the caching. I don't know what the default for REMFile is. Do you?
I think REMFile does not cache by default, but
Okay.
if you want to have a cache, there's an additional option you can specify in REMFile. It's on our tutorial, I think,
Okay.
for REMFile. The other method, FSPEC, also sets up a cache that you can use. So if you expect to access the same large chunk of data over and over again, it's a better way. But if you're just like browsing one time through like what metadata and arrays are there, then you won't need that.
Yeah, so there's the streaming tutorial, and there's this section on using FSPEC, and it shows you how to set up a caching file system for those cases where you do want to do the repeated access to the same data. I guess while I'm here, I just briefly wanted to mention, I forget if I showed the PyNWB docs. They're a great place to get started as you're navigating and working with the NWB files in Python. So we have our homepage that gives a kind of brief overview, and then the tutorial section are going to provide, there's a lot of information here, some of which I already talked about in these tutorials. And so this section is just kind of exploring and working with NWB files. And then as you maybe want to work with more specific data types, or as you're isolating what DANDI sets you want to work with, we have tutorials for those. And so that can be helpful for looking at the different data types contained within OFIS dataset, EFIS dataset, et cetera. And then there's also the streaming tutorial, working with data from ZAR, and then some if you're more interested in the writing side of things. All right, I know, yes. So what the trials table defines, each row is an individual trial, and all that's, I think, required is the start and the stop time of that individual trial. So those timestamps could be then used to access, again, all of the other timestamps for all the other data within the file should be synchronized. So it should be able to access your spike sorted data to index into specific trials you might want from that, or your EFIS or OFIS array data, raw data. So you should be able to use it to access all the different arrays. Yeah.
Okay. If you are having trouble signing on to Dandy Hub, I just posted a message on our Slack channel. Please post your GitHub username in reply to that thread, and we will get it approved and troubleshoot it and all. And if you don't have Slack, I recommend you join the Slack, but you can also just come up to me and tell me what your GitHub username is. Again, you don't need Dandy Hub to download data. This is only if you want to use the free computational resources of Dandy Hub. Yes.
And then I know this was the time originally scheduled, but I'll spend like 10 minutes on Matt and NWB if we have time to go through.
Yeah, sounds good.
Okay. Any questions before we move to Matt and NWB? All right, so.
It works. Good, good, good.
I don't know if we're get into this. I had previously installed NWB without any problems in my lab's computer, but now in this laptop, I tried to install it a while ago, and I'm having an issue that I'm not sure really whether it's related to NWB or it's more MATLAB related, but it just says.
Yeah, maybe Ryan will go help with the.
Oh,
Are you working with this notebook for this tutorial, or?
No, I was able to register into Dandy Hub.
Okay.
But yeah, I mean, as I said, I never used Python in my life, so it wasn't going to be very productive. But I sort of followed through and understood.
Okay. Yeah, and for this tutorial, we'll be installing it from source, so maybe that would work better for you. I'm not sure what installation method you were using.
Yeah, okay. Yeah, I was using MATLAB's default like add-on.
Okay.
oh.
Yeah, I would say if you want to try this and see if we have any issues, this might be more error-proof. All right, so Matt NWB, this tutorial is very similar to the PyNWB one. I'll just mainly be pointing out some of the differences that you might see if you're working in MATLAB versus Python. And so again, in that original notebooks folder I had shared, you can find this tutorial. A very similar one is on the Matt NWB documentation. And so let me actually point that out really first. If you search Matt NWB docs, we should have the Matt NWB documentation has been really recently updated and revamped. So if you looked through it before, you might want to take a look again. There's some great tutorials and examples there. And so again, you can look through tutorials similar as in PyNWB, and then you can also see further documentation about the individual functions and APIs here. All right. So switching back to the Matt NWB notebook, again, to download data from the DANDI Archive, we'll be working with that same file that we were previously. So if you're within the same folder, you should already have it downloaded, but that subject P11 HMH file. So if you go to the DANDI page and then click the folder name and click the download button and move it to this folder. And then to install Matt NWB from source, you can run this section of the notebook. I've already installed it, so it's going to give me an error, but hopefully that should work for Matt NWB installation. It will require that Git is on your path, though. So one thing that's a little different from Matt NWB and PyNWB is that Matt NWB will automatically create the API classes based on the schema. So for most NWB files, these classes will be automatically created whenever you call NWB read. This particular one that we're working with is a little older, so we'll have to manually generate the classes and make sure they're generated for the correct schema version. And so what we'll do is we'll add mat NWB to our path, and then this generate core method that you don't usually need to call. But in this case, since it's an older file, we'll call it to generate the API classes. And then usually all you'll have to do to read an NWB file is to do this nwb.read and then the file path. In this case, we'll add the ignore cache argument since we already generated those classes. And so when you do this NWB file that you get as an output should give you a lot of information about the file. You might notice this will look a little different than the version we saw in PineWB that does a little bit of API magic to make when you're exploring things, but here you'll see what's more representative of what's in the actual file. You'll have your general lab information, keywords, the session ID, start time, etc. And let me make this, is this big enough? Okay. So once you have this NWB file class object, that's going to be the root object for the NWB file. And there are some nice capabilities. If you want to search through the NWB file, you can do nwbfile.searchFor method. And so let's say we wanted to search, for example, for that optical series that we had been looking at earlier in PyNWB. You can do search for, and you get this optical series map. And this will give you information. This will show you that there's this stimulus presentation optical series object within the file. If you also want to search, for example, for all the time series that are contained within a file, you can do the include subclasses and search for all the time series. So we'll see there's some in events, experiment IDs, and then that stimulus presentation. You can then pair that with the NWB resolve method if you want to retrieve these objects that you searched for. So this can be an effective way to query the NWB files. Another way to explore that's MATLAB specific will be this util NWB tree function. And so you can use that. And similar to the Jupyter Notebook representation, we'll be able to kind of interactively go through the NWB file and look at the different components. So let's take a look at the similar stimulus presentation data we had looked at before. So this is going to be an nwb.stimulus_presentation. And we'll see again the stimulus presentation object that's an optical series data type. And if we want to retrieve this object, we can use the get method. And so this is a set that's going to, similar to the Python dictionary, store all of the data objects within the stimulus presentation folder. And here we'll have nwb.stimulus_presentation get, and we'll get all of the information for that optical series. So similar things like field of view, distance, array, and the data and timestamps. So yeah, like we mentioned before, the optical series in this case is storing information about the stimuli that we presented to the subject. And so if we want to look at the data, we can do nwb_file.stimulus_presentation, get stimulus presentation, and the data. And what you might notice here is that this is returning a data stub in this case. And so this is because this is a way to read in the data lazily in mat NWB as well. And so instead of reading the entire data into memory, this provides us this way to selectively slice or index into the data and only read sections at a time. And so if we do want to access the data, we can take the stimulus image data stub and then access it similar as you would into any MATLAB matrix. And one other important thing to note is that in mat NWB, the dimensions are going to be flipped as to how you would see them read in PyNWB. So here we have the RGB first, then Y, X, and the timestamps. And so this can be a little tricky if you're working with arrays in NWB, just something to be aware of. And so then if we look at that image object that we've created, image variable, we can see that here now we're actually reading in sections of our data from that data stub. And then with a little bit of manipulation, we can then display this image, and it's the same one that we saw previously in PyNWB. And if we want to ever read in the entire array, similar to what we did with the brackets and the colons in PyNWB, we can do .load, and so that will read the entire array into memory. In this case, we're reading in the timestamps. All right, and then for working with tabular data, we can use, for example, our nwb.units table. We can use the getRow method to get the first unit of this units table, and we can use that to also access things such as the spike times. And in this case, I'll show another demonstration of aligning the spike times to the stimulus presentation. And so here we have the option to specify, we'll take a specific unit that we want to look at, a window we want to look before and after. We'll get the row of that specific unit, and then we'll get the spike times column and read that in. And then here we'll just align those to each stimulus time and compile it into a cell array. And then hopefully that'll work. Yeah, we'll plot it into just a raster and then a histogram of the spiking activity relative to the stimulus onset. So yeah, that's kind of a brief overview of getting started with MAT NWB and interacting with the data, and then PyNWB and interacting with the data in the first half. And I also showed you downloading data from DANDI with the DANDI CLI, the web user interface, and the Python API as well. Any questions about any of that? Cool. Then I don't know if we have a break or I'm handing it off to someone else.